A healthy immune system operates with strict precision. In cellular homeostasis, innate defenses neutralize pathogens and clear debris without damaging the surrounding tissue. But this environment is under constant pressure. Routine cellular respiration and exposure to environmental toxins continuously introduces reactive oxygen-nitrogen species, or RONs, into the cell. When these free radicals accumulate past a critical threshold, they trigger a defensive response. The cell releases pro-inflammatory cytokines, specifically tumor necrosis factor alpha and interleukin-6, to manage the stress. These cytokines provoke the production of even more free radicals. The initial defense mechanism becomes a mutually amplifying feedback loop between oxidative stress and continuous inflammation. Without a biochemical intervention to interrupt this cycle, the innate immune system's reliance on oxidative defense transitions into a self-sustaining engine of chronic cellular damage. Zingiber officinale, or ginger, operates as a dense botanical matrix of bioactive phytochemicals capable of targeted genetic regulation. The four core phenolic agents, gingerol, shogaol, paridol, and zingerone, are precise transcription modulators interacting directly with cellular receptors. They break the disease loop through a simultaneous two-pronged attack. They upregulate the endogenous NRF2 antioxidant pathway while suppressing the NF-kappa B inflammatory cascade. Ginger's efficacy depends on reprogramming the specific genetic transcription factors that sustain cellular dysregulation. This flowchart maps the pathophysiology of oxidative stress. It begins during cellular respiration, where the enzyme NADPH oxidase generates a highly reactive free radical called superoxide. Endogenous enzymes quickly convert the superoxide into hydrogen peroxide to stabilize it. If this hydrogen peroxide is not neutralized, it reacts with transition metal ions like iron through the Fenton reaction. This transmutation creates a hydroxyl radical, an intensely reactive molecule that tears through cell membranes and proteins. These hydroxyl radicals directly strike and activate NF-kappa B, a primary transcription factor deep within the cell. Activated NF-kappa B triggers the rapid release of inflammatory cytokines. These cytokines signal the cell to produce even more reactive oxygen species, routing the process back to the start. This bidirectional oxidative inflammatory loop perpetually feeds itself, driving progressive tissue damage. Fortunately, the cell contains a powerful internal defense mechanism to break this destructive cycle, the NRF2 transcription factor. This diagram shows how specific ginger phenolics, namely 6-gingerol and paridol, act as biochemical catalysts. They dock into receptor sites, triggering the activation of NRF2. Once activated, NRF2 translocates into the nucleus, binding to the DNA to spark the synthesis of three defensive enzymes: superoxide dismutase, catalase, and glutathione peroxidase. Superoxide dismutase isolates and dissolves the initial superoxide radicals generated by cellular respiration. Simultaneously, catalase and glutathione peroxidase neutralize the hydrogen peroxide threat, preventing the damaging Fenton reaction from occurring. This enzymatic response restores the cell's glutathione balance. Clinically, this is verified by a measurable drop in malondialdehyde, a primary marker of cellular degradation. Gingerols and paridols reprogram the cell to manufacture and deploy its own enzymatic defense arsenal. The second front of ginger's biochemical attack targets the inflammatory signaling node at the cell membrane, governed by the PI3K/AKT pathway. 6-shogaol and zingerone act as primary inhibitors. They physically bind to and block the receptor sites on this specific pathway. Blocking PI3K/AKT cuts off the intracellular signal cascade, preventing the downstream activation of the NF-kappa B transcription factor. Keeping NF-kappa B dormant denies the transcription of inflammatory enzymes, specifically INOS and COX-2. Without INOS and COX-2, the cell cannot synthesize nitric oxide or prostaglandin E2, leading to a direct reduction in localized swelling. This mechanism modulates the balance of T helper cells by suppressing the release of Th1-type cytokines like interleukin-12 and interferon gamma. By shutting down the NF-kappa B transcription factory, shogaols and zingerones actively starve the inflammation loop of its core signaling molecules. The concentration of these phenolics is heavily dictated by the genetics of the botanical cultivar. Elephant ginger is common in culinary applications due to its size, but yields the lowest essential oil and phenolic content of major varieties. This chart compares that profile to red ginger, which possesses a vastly superior monoterpene density, yielding high levels of active 1.8 cineol. This dense chemical matrix results in the highest antioxidant scavenging capacity among the tested varieties. Clinical efficacy is also determined by post-harvest processing, such as drying or cooking. Raw, fresh ginger is dominant in gingerols, which are the primary drivers of the NRF2 antioxidant pathway. When intense heat is applied during the extraction process, the internal chemical matrix undergoes a rapid thermal dehydration. The application of heat structurally forces 6-gingerol to shed a water molecule, transforming it into 6-shogal. An inverse aldol reaction split can also yield zingerone, shifting the extract's primary affinity toward the anti-inflammatory NF-kappa B pathways. Achieving clinical efficacy requires matching the exact ginger cultivar and processing method to the specific biological target. We can now map this entire mechanism back onto the dysregulated pathophysiological loop. The dual-action intervention strikes both nodes simultaneously. Gingerols and pyridols activate the NRF2 antioxidant defenses, while shogals and zingerones block the NF-kappa B inflammatory signals. Attacking the disease loop on both flanks produces a systemic cascade. As reactive oxygen generation halts and inflammatory cytokines recede, the cellular microenvironment is forced back into a stable homeostatic state. Ginger functions as a sophisticated reprogramming agent for the cell's own genetic transcription machinery. Translating these molecular actions into clinical practice requires moving away from generic extracts toward precise phytochemical standardization. Future pharmaceutical development must account for bioavailability hurdles and the precise ratio of gingerols to shogals. Practitioners and formulators must deliberately match the botanical variety, like red ginger, and the preparation state to the desired biochemical outcome. Zingiber officinalis serves as a frontline tool in managing immune-mediated and inflammatory diseases through precise molecular targeting. True immunomodulation utilizes nature's chemical architecture to restore cellular homeostasis at the transcription level.